Okay.
Does it show it's recording?
Yes.
Cool. There we go.
All right.
So now you guys are all like YouTube famous.
Yeah, yeah.
No, I'm just kidding. It's going to be an unlisted link that I use for YouTube. So the only way you guys become famous is if y'all share the link. All right. Good catch. So we start with the intangible in this class. And so we're in A&P. I could show you blood. I could give you a femur. It's really hard to give you an E. coli and show you what it looks like. Rest assured, you would know if you had an E. coli. True to form, I have terrible luck. My water system in my neighborhood in Orange County broke, and it's been on the fritz, I would say, since the pandemic. And so what that means is we had a chlorine pump that's meant to sanitize the water, which comes from the ground aquifers, fail. But when pumps fail, they tend not to just go all at once. They overchlorinate and then they underchlorinate, and then they overchlorinate and then they stop working. And so I probably have been battling an E. coli infection or at least some sort of extra intestinal microbial infection since March. And I finally had to deal with antibiotics and a CT scan which is scheduled to try and figure out what was going on. So rest assured, I can't hand them to you. I can't show them to you without a microscope. But despite their diminutive size, they pack quite a punch. And so in water, we'd like to see clear water, but unfortunately, even coming out of our tap, there's a biofilm in that water where you probably have some bacteria. They probably don't bother you so much. There could be some viruses in there, but they're probably not human or animal viruses, hopefully. So we kind of overlook that. Fungi is an interesting one. We know there's macroscopic fungi, the sexual products. So toadstools and cup fungi and magic mushrooms and all that good stuff. But there's a lot of fungi or members of the kingdom fungi that you don't see. The hyphae, the mycelium, candida on the surface of the skin, right? We only notice these things when there's an overgrowth. And I know that we really hem and haw about bacteria, viruses, fungi, but when we hem and haw, it's about the 2% that cause disease. It's really not about the other 98% that have an ecological role, a food microbiological role, something that they're doing that's useful to the environment or for us. Fungi kind of fall into that category. If you've ever had a tinea, if you've ever had yeast, it's annoying. I just had 2 toenails pulled out the other day for an unknown reason other than the nails were shot. And it's frustrating because you know there's something there and you can't see it yet. An overgrowth is always visible and makes itself known. So we talk a little bit about fungi. Protozoans get interesting. Hopefully you guys haven't had to deal with things like Giardia or that brain-eating amoeba that we've got in the south of the United States, Texas. So hopefully your pools are chlorinated, the lakes are stayed out of, or at least you wear nose clips. Some of those protozoans are pretty savage. Helminths bring us to worms, and in the US, you still get worms. Children get pinworm all the time. Hopefully parents, aunts, uncles, you know about pinworms. They like to get into the digestive tract because children put everything in their mouth and they crawl out of the anus at night. They burrow into anal tissue and they lay eggs. And the characteristic behavior is the kid with his or her hand in their pants, itching at their anus because the worms are doing what they do. Have no fear, a little vacuuming, a hot wash, and a small poison pill, and you deal with those little worms. Right? Mainly, we're thinking nematodes, so the roundworms. We're thinking platyhelminths, flatworms, or ashy helminths, if you want to think of them that way. And trichinids and hookworms all fall under those categories. More recently, though, in the U.S., we've seen a lot of worms that end up in other places like the eyeball. And those have been primarily centered on the West Coast and Texas around the river and cattle industries. And so what you end up finding is your eyes start to swell. You may actually see the worm swim in front of your iris. And obviously any discomfort, swelling, or redness in the eye is grounds to go see an eye doctor immediately. The last piece of this is algae. And again, algae are an interesting piece that falls under microbes. Some algae, like the cyanobacteria on the right, could be in the kingdom Monera, while algae is typically used in context of protista. Things like Volvox or Spirogyra or Oscillatoria, Aphasimenon, Microcystis, those types of things. Most of them are harmless. They're food crops. We use them in drinks like chlorella all the time, but sometimes they do cause injury to humans and the ecosystem in the form of blooms, right? So they're in the cells, they're in the water normally, they're in ecosystems normally, but something happens, pollution, acid rain, and it releases the normal condition, and they grow and they become numerous, 10 to the 6 cells per mil. And some of them produce toxins, neurotoxins, digestive toxins, ciguatarotoxin in the Caribbean is another bad one. And some of them are poisonous to the point where, yeah, you could die. So while, again, totally small percentages, these are things that often we ignore. Often you don't see them with the naked eye unless it's a clear overgrowth. And that makes the studying of these a little different and a little more challenging than you had been used to in A&P. Doesn't make them any less cool. In fact, I kind of like this much more. Sick people, microbes, well, they don't talk back. They're much more interesting. I would have made a bad doctor. Oh boy. So aside from just looking at the growth of these things, and a classic microbiologist wants to grow them. They want to grow them in the lab. They want to grow them in the lab azenically, right? And so when we say azenically, that means pure culture. And so E. coli or staph or any of those human pathogens, yeah, they culture very well purely azenically. With food microbiology, with soil microbiology, it's sometimes very challenging, environmental microbiology, to get something to grow azenically. Because they live in microbial communities, I might produce the histidine that you need, somebody else is producing the arginine that I need. And so if I separate that community, then my metabolism is incomplete, and I'm not able to grow on my own. So that's kind of an interesting thing. That's the main focus and main study of classical microbialists. Some of the modern microbialists, though, have a lot more interest in a lot more fields. Immunology, it's a huge deal, right? If we use COVID as an example, the flu as an example, some people are utterly destroyed and ravaged by these viruses. Some folks are asymptomatic or they get a very mild case. We learned from the 1918 pandemic, it was the body's response that really decided if someone was going to survive or not. And we're seeing some of that today. Obviously, with things like HIV, we know how important the immune system is. And immunology is a great branch to follow up on after microbiology if that's something you're into. Complement cascades, T cells, B cells, NK cells. It actually is fascinating. You might choose to go into public health or epidemiology. And it's a little bit of a different approach where you're looking for cases of strange illnesses above a background number. You start doing contact tracing. You start trying to figure out where it came from, or sometimes where did it go? Ebola, great case of epidemiologists. Or even, let's say, you eat out when we can, when it's safe, and you get sick after you've eaten out. You probably call your local health department, and they're going to be responsible for following up with you. You're going to be asked to provide a sample. Rest assured, that's fun. I've had to do that twice this year. And they're going to inspect restaurants, and they're going to get to the bottom of the case of your illness. If you've got time, I know you guys don't. There's a good podcast. I'll try to dig it up. It's called The Ghost Well. Maybe you guys have read it in nursing or heard about it in nursing. It's about England and a problem with cholera. And the short version is there was a well, it was contaminated. When they took the handle off, the disease went away. And the early epidemiologists knew about this because they looked at the incidence of cases, they clustered around the well, they made a hypothesis, and they saw it through. So public health micro is actually very cool. Food, dairy, and aquatic, it's funny how when we package salad dressings, when we process meats, how many organisms things come in contact with. And so some scientists make sure that harmful, extremely hazardous listeria stays out of your food by doing routine testing and shutting down lines. Dairy, right? Pasteurization, it's key. There's a return to whole milk movement. I don't know how you guys feel about it. I don't know how you don't feel about it. But what people would like is for milk to come from the udder to a bottle that you can purchase. And of course, with the way we handle dairy cows, their udders are usually raw, they're bleeding, they're pussy. And so the pasteurization process protects you from that. It also destroys some nutrients, and that's really where the debate is. Aquatic micro is just like it sounds, especially as more and more of our food sources are farmed, especially if you're a fish person. It's more important to keep the fish healthy and get rid of the microbes that could make them sick because of a process called bioaccumulation. So if they eat the microorganisms that are toxic and then we eat the fish, obviously we're eating the toxins and nobody wants to deal with that. If you're bored, there's a good, super fun site right off of Cold Spring, New York. It was so contaminated with cadmium, instead of bioaccumulating, everything just died. And so in Foundry Cove today, you find one species of worm. They're called polychaetes. And even if you bring in similar species from other spots, they do not fare well. So they acclimated and they have, through the change in frequencies, adapted to live in that ecosystem, which is harsh as can be. The biotech and the engineering kind of go hand in hand. And this is really an interesting debate. So the pros could be, now we make human insulin from E. coli. We don't have to rely on horse or other animal derivatives. We make factor VIII now just by having a gene, right? We can help people who are missing that. We can make foods that are resistant to pests or to pesticides. And I'm not going to say all of that is positive, but I think in some cases it can be positive. Recombinant DNA technology is an interesting one. Viruses have been doing this for years and years and years and years. It's called lateral gene transfer, laterally transferring genes from unrelated species. Viruses are masters at that, right? We can learn to do this in the lab. I hope there's an exercise for you guys to read up on antibiotic resistance. But to the folks who have any antibiotics left in your cabinets, right? So if you open the medicine cabinet and you find something in there, you all know by now that you shouldn't, right? Take till the end, take as directed. But you guys are forcing antibiotic resistance and the exchange of gene information by applying random selective pressures. So all of this really folds back into, we're studying things that can't be seen with the naked eye. And all of these things could apply to viruses, fungi, bacterium, other eukaryotic pathogens, and the host response. So there's a lot of cool things that we can look at once we start to crack that book and realize all of the things that fall under the umbrella here. And obviously, I don't need to name them all. You guys can kind of see how there's a lot of harmless stuff that we can do. And I wish we had time to really take field trips and look at this stuff. Biosynthesis, mineral cycling. I mean, this is like classic oceanography, classic aquatic ecosystem. Decomposition. I would love to be able to go to Chernobyl and see the forests that fell almost, 30 some odd years ago now, that still haven't decayed. They've done some amazing experiments with garment bags. The further away you go from the site, the more microbial degradation of dead and decaying matter there is. Close up to the site, it's still too hot, and all of those organisms have been eradicated. Drug discovery, food production. I love cheese. And there's some amazing cheeses out there, right? Lactic acid, fermentative products. Maybe some interesting ones. I don't know how adventurous you are. But there's a good Italian cheese that comes from Sardinia. And if you know casu marzu, it has worms in it. And I shouldn't say worms, but they're maggots. And the maggots eat the cheese, and their leavings are enzymatically different than the products that went in. It produces a stench, but it produces apparently an awesome flavor. Vinegar, I can distill it all day long. It tastes like garbage. There's something called the Orleans method. You use a mother, it actually distills it using an organism rather than a still. Amazing. Wine, sauerkraut, all the stuff, fermentative products. And obviously, there's a lot of organisms that make drugs, right? Penicillium makes penicillin. Grisia fulvum makes Grisia fulvin. A common sea sponge is the source of Abreva. Right? Who knows what kind of trees and other medicine plants are going to be in these rainforests? Or who knows, maybe even your backyards. If we took the time to study them, to make the proper extracts, and I hate to say this, but it's true. Everything that we're going to learn from the legalization of marijuana can be applied to so many more things than just recreational use. And I think you're going to see botany take off again. And I think you're going to see the microbes that are associated with this stuff take off again also. We already know about change in ecosystems, composting, bioremediation, Bioturbation is interesting too, the activity of animals that affects the decomposition of materials and redox and chemistry boundaries in nature. All of that, though, is probably not why you're here. And it's too bad because it's interesting, but I'm sure most of us are here for the health professions and the idea that there's a link between microbes and disease, right? Better known probably is the germ theory of disease that we'll attribute to Dr. Koch in just a minute. But that unfortunately is the negative, at least in the human factor, right? The fact that there is disease that causes morbidity or mortality. Unfortunately, in the grander scheme of things, everything that's produced eventually has to be decomposed and remineralized. So we are really just part of the cog in that ecological wheel if we take the human element out of it, which I know sometimes is hard to do. Pathogens, we're probably going to define as organisms that would cause disease in a healthy individual. So I have staph on me right now. You guys probably have staph on you. So that particular strain is probably commensal, and it's obviously not causing disease. I start consuming my water, and the E. coli get into me. Well, that E. coli becomes numerous and causes disease in an otherwise healthy individual. Or you go out to eat, and something's contaminated, and you guys are otherwise healthy probably, and you come down with something. So we're really talking about virulent microbes, although there are opportunistic pathogens that tend to affect those that are susceptible. Or sometimes by accident, you clean your nose and you cut it, and one of those staph gets in there. It shouldn't have been in there. It's going to cause disease. For our purposes, we're thinking about 2,000 different microbes. And so 2,000 different particular species from a particular genus. I don't mean all the strains and all the subtypes, but that's a lot of stuff. There's 1 billion infections a year worldwide, give or take. This year could be a little higher. And deaths from microbial infections are in the tens of millions, 1.3 times 10 to the 7 per year worldwide. Some of it is the organism grows. Some of it is the organism's toxins that they produce. Some of it is just the mere presence of the organism. Gram negatives, for example, have a sugar-fat combination in their cell walls. And just the presence of this in the bloodstream is immunogenic. So it causes inflammation and it causes cells to rush to the scene. And it's kind of interesting because gram positives behave a little bit differently. Sometimes, the pathogens do their thing by opening up a wound and secondary stuff gets in there. So they really have evolved an interesting biomechanical mechanism to make this happen. Urinary tract infections are probably one of the most common reasons why women go to the hospital. And they're pretty easily treated normally with antibiotics, except now we're seeing a lot of resistant ones also. And so a case of Klebsiella pneumoniae ordinarily would be a 7-day sentence on antibiotics. Sometimes it's a little bit more than that now.
I'm sorry, Professor.
Have at it, go ahead.
I apologize for interrupting, but I have a family emergency. May I leave class early, please?
These are going to be recorded, so if you gotta go, go ahead.
Thank you so much. I apologize.
All right, no worries. Definitely we have to have family first. Right, so these microbes are really interesting. And the reason why the Klebsiella, or the reason why Helicobacter pylori get into you and cause disease is because they have special biochemical pathways. In both of those cases, H. pylori, I'll just kind of throw that in here, and this should be in italics, shame on me. Which it's not going to do for me, unfortunately, in the chat. Abbreviate that. Have what's called a urease gene. And so what they do when they get into your stomach or when they get into your urinary tract is they start to produce a compound known as urea. And urea has a pH of 11. And so your urinary tract is 5. When you mix 5 with ammonia, it goes up. It becomes more alkaline and you allow more things to grow without being killed by the acidic environment. In your stomach, right, we've got acid, pH 2. You add ammonia to that and you raise the pH to 5 or 6, where the Helicobacter can take hold. And apparently, if it lasts long enough, it actually can spread even into the duodenum, where you can have peptic ulcers. So without that urease gene, these organisms wouldn't be anywhere near as effective as they are currently with that little extra arsenal tool there of enzymes. And we can see a bunch of stuff in terms of infectious disease or diseases from microorganisms. COVID isn't on here. This is an older one, but certainly it should be. HIV is on here. Diarrheal diseases are still very real. TB, believe it or not, is a 1 in 3 in America. Think about that. No, not America, in the world. And so if you look left and look right, or in my Brady Bunch, if I look up and down, if the one in the middle, no, it doesn't work that way. The US is very good. We test, we screen. But most of the TB that we see in the US is derived largely in prison populations. And that's pretty much what you see around the world. So the larger the prison population, probably the greater numbers of TB that you tend to see. And a lot of that is centered in Russia. If you look at the gulags, you see a lot there. It's a smart disease. It's a very stubborn disease. And it's amazing that after however many years of the miasmatists and then the gaunt Renaissance period, tuberculosis is still a scourge. What else is in here? Measles. My dad had measles twice. Don't ask me how that happened. The treatment for measles is very different, but we do have the MMR these days. Hepatitis B, hepatitis C, Hep A, they certainly contribute. And so we are far, far, far from a golden age of microbiology where we have most of this in control or vaccinated, or at least we have drugs that work. I can't say that we do. In some of these cases, at best, we're managing it. And for the folks who like conspiracies, I am totally one of those people. You have to think, are there drugs that work available and we're just not getting access to them? Well, certainly in the case of President Trump, we know that there's drugs that exist that we don't have access to for cost reasons, but they couldn't use anything on him that they didn't want disclosed. And so that also means that the pharmaceuticals who are out to make a profit probably aren't going to hide a cure. Although there is that conspiracy speak about the cure is almost always cheaper than managing symptoms. And so why would we want to fix that? Unfortunately, that's glib. I don't know that I prescribe to that. And certainly I don't have any direct evidence of that. But I'm hoping that as we proceed with microbes and as we proceed with DNA technology, we already have a Jason Bourne-like fix for some diseases like hemophilia. You can get a shot, it changes the DNA of the cells in circulation, they make factor VIII for about 18 months, at which time your body has cleared all of the changed cells, and you would go and get another injection. Those are in trials. There's another 8 or so, including one for HIV, that are in the works. As you guys know, there is a genetic mutation that gives you somewhat protection over HIV. Magic Johnson is one person who has it. But it really is a deletion of the viral co-binding sites. It's nothing to do with the money. Just got to say that out loud. It's about the binding sites that allow the virus to dock. Everybody's got the CD4. Not everybody has the CRCX5 and the CCR4, or CCR4, CRCX5. There's 2 accessories. So we're making strides, but sometimes it feels like 2 forward and 1 back, or 2 back and 1 forward, depending on the day. Microbes could have all types of appearances. The prokaryote on the left, that's chapter 4. We'll look at those in depth. No nucleus, no membrane-bound organelles. There is a cell wall, though, which makes the prokaryotes distinguishable from their cell wall. Gram-negatives, gram-positives, certainly acid-fast organisms all fall under that category. Or eukaryotes in the middle, they look pretty typical to the cells you've seen. And you're thinking basically protistas, helminths, fungi. On the right is something kind of interesting, and those are viruses. And there's 2 different kinds here that I'm showing you. The one on the right, the red one, the skeletonized version, is a bacteriophage, and those bacteriophage only affect bacteria. There was once upon a time a theory where I could infect you with a phage. If you had a staph infection, the phage would clear your staph up. And allegedly, in parts of the world, the former Soviet republics continued on this research after the fall of the Soviet Union, and the Ukraine actually has a stock of virus for things like MRSA, supposedly. The other virus you see is an enveloped virus, and it's an animal virus. In order for a virus to affect animals, it has to be enveloped. Otherwise, it would never make it past the cell or the immune system. And so things like HIV and influenza and coronavirus have the spikes that you see before you. And those spikes allow them to get into the cell. And sometimes those spikes allow them to get out and become a budding virus without actually lysing the cell. Envelope viruses affect all animals. Non-envelope viruses typically don't do anything other than raise an immune response. And the bacteriophage on the right are literally only affecting bacteria. And so when we talk human viruses, nobody should ever think of the little creature on the right, although functionally they work very, very similar to one another. So it's basically, we'll learn it once and we'll sprinkle the details, because for eukaryotes, we use a few different words. Obviously, size is not an issue here. I always ask, does size matter? People giggle and laugh, but honestly, it doesn't. You know, that louse or that bed bug still wreaks havoc, right? That black bread mold at about 1 millimeter, you see, you taste, you don't want to eat it, and it provides a means for reproduction. You get down to polio at 10 nanometers. Anybody who's ever seen footage of FDR knows polio is a thing. It works even though it's tiny. And certainly those of us living in the shadow of Indian Point know that hydrogen atoms and molecules at the atomic level are just as dangerous and powerful as the rickettsia or a staphylococcus. So even though we can't see these things, they're diminutive, they're small, they're hiding, they're invisible. They still pack a punch, don't they? Let's see. So that's kind of a quick intro. There's a couple scientists that are worth talking about. And again, it's a nice walkthrough just to kind of think about how we got to where we are today. It's unfortunate you guys can't get your hands on the scopes in the lab. We do have some nice binocular microscopes with oil immersion lenses that I purchased a couple years back. I don't know, we'll call them in the $800 range. The first microscope, though, definitely would have been less advanced than Fisher-Price, although any place is a good start. And Anton van Leeuwenhoek was really the modern inventor, only held up by the advent of, strangely, the ability to polish glass. And so once we were able to make a concave shape and kind of curve glass, he was able to produce a microscope that he's holding in his left hand that's pretty simple, basically a metal plate, a pointer, and a lens, where you put your sample on the pointer and held it up to the sky. And you could imagine this poor man's surprise when he went down to the river, the stream, the lake, that was his drinking supply, of course, someone else's bathroom, and he saw things that he couldn't see with the naked eye in the water, things that were moving, things that might have been dividing before his very eyes. I don't know about you guys, in the 1700s, late 1600s, I would have freaked out. Right? If you did that today with your water at home, y'all probably still freak out. Let's not even ask when the last time you changed the filter in your refrigerator was, or cleaned out the bottom drawer. We probably don't want to know. But in the drinking water, it hits a little different. And so he looked and he found things that were moving, things that he didn't think should be there. And even though he got up to 300x magnification with his very primitive scope, it was enough to see a world that he had no idea that existed. And really, this started the branch of microbiology. This started the scientific inquiry. This started cell theory also, when you get down to it. Now, obviously, this gentleman produced the hardware, which looks something very similar. But it was Robert Hooke that coined the term cell. And it was others like Pasteur and Reddy that really tied the theory together over the course of roughly 200 years. To the very right, in panel B, those are actual drawings from Leeuwenhoek. And you see a bacillus on top. You see something strange and moving in figure B. You see some cocci in E. We've got a spirochete or spirilla in G. And in the bottom figure, we've got some vibrio. We've got some bent or curved rods. And about the time when these would have been taken or drawn, diarrheal diseases were a big problem. Cholera was a big problem. And we know today that 90% of the cholera patients don't have the toxic strain. They have voluminous diarrhea. You keep them hydrated, you get rid of the fecal material, they recover just fine and no one else gets infected. Unfortunately, we also know what happens when we don't keep things separate. And for those of you who remember, and if you were affected, I apologize, there was an earthquake, a severe one in Haiti at the latter part of the OOs or the early part of the 10s. And there was a massive wave of cholera that swept through the island nation because the relief workers brought the cholera with them, and the sanitary sewer system bled into the drinking water. Right? And so even in, we'll call it 2010, with sanitary conditions, they fail, and the lessons learned from the past are as valuable as they were when we learned them, right? And that kind of goes hand in hand with COVID. The lessons that we learned from the pandemic of 1918 are still every bit as valuable as they were then now. Mask wearing, isolation, distancing, hand washing, right? You can't beat the basics. Vaccines are seen as a good thing generally, but maybe seen as a bad thing. And I think the bad ones are because of medical mistrust. That article that was published in the late 1990s that talked about thimerosal and autism was really irresponsible. And as most of you know, that paper was retracted. The person who wrote it was stripped of his medical license and title and was jailed. The reason being they didn't follow the scientific method, and they fabricated data to fit the narrative they wanted to tell. When everybody got the paper and they read it and they tried to find the same data, they found that they could not. And so he was called a fraud, and he admitted to making up the data, but we did damage for decades. Vaccines aren't new, by the way. Vaccines go back to the 1700s. And back then, methods were crude, and biological samples were often shared, but they had the effect of doing the job, and immunity was indeed produced even from these crude primitive vaccines. Modern vaccines are typically heat attenuated, so sometimes there is live virus like the polio vaccine, or dead virus, so it's heat killed, or it's a fraction, a piece of a virus using maybe a different virus as a vector, or now even mRNA for the viral proteins is being used. The first time we diddled with this and we used something unnatural was the hepatitis B vaccine. The first genetically engineered vaccine to hit the market, 1994, give or take. Probably went back, obviously, into the 80s, as most vaccines take 10 or so years to fully develop under normal conditions. And so the Hep B vaccine was a big step in both protecting against hepatitis and ushering in a new age of vaccinations. And while there is mistrust, I do have to keep going back to the 1700s when we had a cure, a vaccination, a therapy for smallpox that a lot of people kind of ignored and forgot. And we had to reinvent it in the 1900s when people had a little more faith in science and medicine. And so if you've heard the story, I'll be brief. Basically, Jenner had noticed that something funky was going on in his milkmaids. And he noticed they had cases of cowpox, but the cowpox provided enough immunity against smallpox. Smallpox is thought to have originated in South America, perhaps, as a sexually transmitted disease, as a skin-to-skin contact disease, and it was brought back to the old world where it became an STI. And at its height, smallpox probably killed 30 to 40% of the people who were infected. The disease goes back to the pharaohs. You can see Tutankhamun probably had smallpox. And so obviously getting rid of this was a big deal. The milkmaids got cowpox from handling the udders, but they never contracted smallpox. And so he started to grab the scabs of the cowpox and later the scabs of the smallpox, grind them up and use them as inoculant. It worked. Certainly wouldn't pass the FDA or any government standards, but back then we didn't have them, but it worked. So probably the father of vaccinations is who we're seeing in this picture on the left. And as we're also aware, the World Health Organization, by going to every nook and cranny and corner on Earth, declared smallpox eradicated, give or take 1977. And because there is no other reservoir, there is no place this virus hides other than humans, and unfortunately some freezers in some places, they called it eradicated. It's no longer in existence. We still vaccinate the military. We still vaccinate in the case of people thinking they were exposed due to terrorism. But otherwise, the last probably vaccination of a common person in the US was probably 73, 74, 75. If you're at the supermarket in the summer, though, and you happen to notice somebody with a small circular scar on their upper arm, chances are it's a smallpox vaccine where they take a forked needle and they stick you probably 20 or 30 times, or they're from a different country where they vaccinated later. And people sometimes talk about it. They're good stories, but they did the job, right? We got it out of here. And now we'd like to use vaccinations to get rid of some other things. It worked here because there's only one reservoir, humans. Polio, we tried. We messed that up big time because the only other reservoir besides humans is what? Anybody know what it is? Give you a hint. It's where heparin comes from. No, it doesn't help. The pig. The pig is the only other reservoir that we know that carries polio. And when I say we messed up, we did. We were providing vaccinations that had type 1, type 2, and type 3 polio in them. We stopped vaccinating against type 2 polio. And when people started to shed, because people shed polio when they're vaccinated, there was a biochemical change that led to type 2 polio being results, and infections of polio blew through the roof. And they call it vaccine-associated polio. Obviously, any polio is no good. So we've definitely undermined some faith in some of the developing worlds, and it's going to be hard to earn that back. Here we vaccinate everybody. It's that good cherry liquid. And since we've got the herd immunity, nobody really shows signs of polio infection anymore, right? And we'll get to when that actually started. How about spontaneous generation? How do you guys feel about this? Right, it is the third tenet of cell culture, and the cell theory, right? The cell is the structure, the unit of structure and the unit of function, and omnis cellula e cellula. All cells come from pre-existing cells. So spontaneous generation was a really interesting one. And the two stories basically are ready with the meat and Pasteur with the broth. All right, and I'm sure you guys have seen these before. So what Pasteur did was he had a broth container, really very much like the turkey soup you guys might have for Thanksgiving. And as you know, when you put that vat of soup into the fridge after you're sick of eating it, usually the garage, you forget about it, and you tend to remember it when you smell it. And so that turkey soup is really a prime broth for microbes. It's like one step richer than the LB or the nutrient broth we'd use in the lab. And so what he did was he took and boiled some broth, and he left one of them open to the atmosphere, and he put a neck on one of them, very much like the trap on your sink. And that trap is designed to catch particles, and that's exactly what the trap did in his experiment. It trapped small microbes and small cells, but it allowed air to be exchanged. And he demonstrated that it wasn't air or magic that was causing the broth to spoil and become cloudy. He showed it was actually microbes that got caught in the trap. Reddy did the same experiment with beef. He put beef in a jar, he put a lid on it, beef in another jar, he left it open. He showed there were maggots on the beef he left open, and that the beef that he closed was perfect. Somebody, though, said, you messed up, son. And they got at him for messing up the control. You can't remove the oxygen from the jar because now you've got two different conditions. And so what he had to do was use cheesecloth, because this is the 1800s. Nylon hadn't been invented yet. And cheesecloth the top. And that's where he actually caught the flies laying the eggs in the cheesecloth, trying to get at the meat. So it wasn't just poof magic, there's maggots. It was the flies led to the eggs, led to the maggots. Hence, cellula e cellula. From cells come other cells. But Pasteur did a lot more than that. He definitely showed that microbes were the cause of fermentation and food spoilage. And obviously, pasteurization is in his namesake. The Pasteur Institute in France was also the place where HIV was first characterized. And Luc Montagnier was one of the first French folks to get the Nobel Prize for his work on HIV in 1992. Probably didn't also know that he worked in concert with Dr. Koch, and he developed the rabies vaccine, right? That was the rabies vaccine that was like 20 stabs in the stomach before we had the abbreviated series. Interesting fact, you can vaccinate for rabies after exposure. It's one of the few viruses you can do that on because it replicates very, very slowly. And for those who know their history, Edgar Allan Poe was theorized to be a drunk, very, very world-class poet. But in his latter years or latter months, he was a stumbling mess. And the theory is now more, he wasn't a drunk. Well, maybe he was, but he died of rabies. And that's the current thinking there. Obviously, the linking of microbes to food spoilage and the linking of microbes to disease is really the foundation of modern microbiology, right? We don't get sick because our hair is wet when we go outside. Well, if I had hair. We don't get sick because I don't wear a jacket or I wear shorts all winter. That's not why I get sick. Right? We get sick because of microbes entering the body. And it took a while to get there. And there are still folks who say, put on your coat, you're going to catch a cold. Bullshit. Right? That's not how it works. Now, does stress run you down? Sure. But you wouldn't get sick if there wasn't a microbe when you were stressed, right? So the work here is super, super important. Now, I don't want to downplay what's on the screen, and I'm just going to go with the last part about poverty, social status. It definitely can put people at a disadvantage to being inoculated with a microbe. I have to say that because that's what the data suggests. In some places, it's not necessarily poverty, but it's the stigma that people have when they're sick, why they don't seek treatment. And one of the biggest things there is leprosy, right? There's still leprosy on Earth. We can treat it with antibiotics very easily. Most people who get it don't seek medical attention until they're already disfigured. And so that isn't necessarily a character flaw, but it's a societal flaw. And so while the microbes still do the work, there'd probably be less disease if some of the things were a little cleaner, if there were less stigmas, and if we had better health systems in many places. The person responsible, though, for the germ theory of disease is Robert Koch, right? A physician who definitely worked with TB. He did some cholera work for sure, and that's actually how Koch's postulates were derived. And he worked with anthrax, Bacillus anthracis, probably the first to characterize the cutaneous versions and the pulmonary versions of anthrax. Now, Koch's postulates are very interesting. They were the gold standard of determining what microorganism caused which disease. And as I alluded to earlier, he was a cholera guy. And so when he was treating cholera, he pretty much had a Vibrio isolated. But it was more than that. So his postulates kind of go like this. Observe a disease and isolate a microorganism. Once you isolate the microbe, you infect a healthy person with it. And you see if that microbe causes the same disease in that healthy person, and then you isolate it one more time. And so what he did himself was he took one of those cholera cot buckets filled with feces filled with cholera, got his thumb in there and inoculated himself orally. The good doctor came down with cholera. He was as sick as could be. And once he was better, he went to the stool samples and he isolated the same bacterium. And so Vibrio cholera was linked to cholera because of his efforts. Today, this raises some severe ethical questions, largely in the case of diseases where we have no drugs or cures. So for example, I'm pretty sure nobody would be giving anybody HIV to prove Koch's postulates, right? That would be wrong. I'm not saying it didn't happen someplace, but certainly that's a failure of Dr. Koch in the 21st century. Obviously, we wouldn't want to give anybody coronavirus. We certainly can isolate the bug from people, and people give it to each other enough, we're pretty sure what it is. But coming from the idea of gnomes and spirits and bad air made you sick to linking it to a disease, this was a huge step in microbiology. Likewise, Dr. Ehrlich, who definitely has some claim to fame, produced the first drug to target syphilis. Now, interestingly enough, this is pre-antibiotics. Salvarsan had a lot of side effects, largely because it was produced with heavy metals. And as the name ars sounds, it was largely arsenic that he was giving people. And so if the syphilis didn't kill you, right, primary syphilis is just a chancre. Secondary syphilis is usually inflammation and kind of underground. But it's that tertiary syphilis where you have bone and skull degradation and neurological impairments. where folks really needed the help. Once the damage is done, getting rid of the microbe doesn't undo the damage. And I remember watching a lot of World War I films where G.I. Joe would literally come into the doctor. The doctor would say, "Hey, Joe, I'm sorry to tell you, you have syphilis." And the guy would say, "Well, I guess that's it for me, doc." And so this was a really big deal in terms of a treatment before we had penicillin. Obviously, there was a crime perpetrated against the American people with relation to this drug and syphilis. And so while we talk about ethics and it can't happen in this country, clearly there have been lapses, and obviously those are terrible mistakes and tragedies. The advent of antibiotics was really a big deal. Heavy metals are antimicrobial, but they do terrible things to the body. Lead, arsenic, and those kinds of derivatives are really, really dangerous. And so while it worked, there were better things. And certainly Sir Alexander Fleming discovered one of those better things. Penicillin, 1928. With Florey and Chain, they made it a medication. They won the Nobel Prize in 1945. And obviously this drug still saves countless lives today. Remember, in the 20s, if you had a paper cut, you might actually die if you got a staph or strep infection in it. Right? If you had a pimple or a boil, you probably were going to die because there was no such thing as Neosporin in those days. It just went unchecked. Remember, the life expectancy in these times was 46. Right? If you were a soldier, it was probably 6 minutes on the battlefield, depending upon where you were. Penicillin was a great discovery, 28. The next one to be discovered was streptomycin in 1945 by a group that you probably have heard before, Avery, Griffith, McCarty, and McLeod. Right? They had something to do with the principles of transformation as well. These are his co-conspirators, and I say that loosely because their work was magic. And unfortunately or fortunately, they tested their work on in the 1940s and the late 1930s with American GIs. So people were shot up. Generally, they would develop infections. They medicinized the penicillium derivatives, they gave it to the soldiers, and they thought so highly of this, they even reclaimed their urine and reprecipitated the penicillin out of it. Right? They thought so highly of it then. Imagine if we treated antibiotics like that today. Streptomycin was produced in 1945. It was already resistant. Most bugs were already resistant to it, tuberculosis resistant to it by 1947. Right? So the antibiotic thing, promising, part of it is how we use it. Part of it is, we have some in our cabinets when we shouldn't. Part of it is it's a war, and these microbes can reproduce every 20 minutes, right? We don't reproduce anywhere near that quickly. So how do we combat the time and change? Better living through pharmacy. These two gentlemen, also extremely important. If you lived on Long Island, if you went to school out there, I can't tell you how many Jonas Salk high schools there are in America. He produced the first polio vaccine in 1952, and it was a heat-killed version. Very interesting thing, especially if you are able to speak to your parents or your grandparents about life before the vaccine. You couldn't go to a mall, a movie theater, the pool, a park in the summer because it was just too crowded. And that's where you got polio. And so a lot of parents kept their kids out of doing things. You had to be very careful or you contracted the disease. The vaccine was amazing, and it changed the lives for between 10 and 30 million Americans at the time. Unfortunately, unbeknownst to us and unbeknownst to Dr. Salk, it came with a slight bonus, and that bonus is a virus called SV40. SV is for simian virus. And so the way the vaccine was produced, simian virus number 40 co-purified with the vaccine. And it was in fact given to that 30-some million American people in the 1950s. And folks often wondered if this was the origin of HIV or something else. And honestly, to this day, we really don't know. And so when you think about the things in vaccines, it's not all rosy. I have to admit that. There are metals and remnants of cells and things in there. And I think for things like polio and COVID and MMR type stuff and tetanus, the risk justifies the means. For things like the common cold or maybe chicken pox, you're getting into that realm of, is it really justified? Is it something we really should take? Lyme disease, there's a vaccine. That's one that kind of you shake your head at, you wonder. Meningitis, though, I don't know. I think the risk kind of justifies the means there. Pneumococcal diseases, probably if you're a particular risk group, it's worth it. But this process does highlight that even the brightest minds and the best of intentions do sometimes make mistakes. And that's just being human. That's fair. What came along after, the gentleman on the right, Albert Sabin, produced the liquid that we consume today. And it's a live but attenuated virus. So that means you count on your immune system to beat it down and build the immunity that you have today. If you're somebody who's immunocompromised, if you're somebody who takes immunocompromising drugs, biologics for like arthritis or Crohn's disease, or have an organ transplant, getting these types of vaccines is a little risky because you may actually get the disease rather than the immunity if your body can't fight it off. Right? So that's why often when you watch the commercials, it says, if you are compromised or you have concerns, contact your doctor. I need to do that with my autoimmune stuff just to make sure that we are okay. And so with this penicillin and the vaccines to date, the 1960s was the golden age of microbiology through the 70s, really kind of perpetrated by the Vietnam War. You had a lot of free love, a lot of free sex, a lot of prostitution, a lot of pillaging with all of the heinous things that come along with it. And so we were creating and finding diseases and passing them around by the end of the 70s faster than we had microbial drugs or therapeutics to treat them. And that's where the resistant strains of gonorrhea started to come from. That's where the resistant STIs with penicillin were seen. And quite frankly, that's where HIV came from. Right? It came probably from plundering and, well, there's more to it than that. But the plundering ceased to be plundering when those people went home and they started to have sex with their wives and their husbands again. And so that's when it finally started to make its way silently around the globe, with the life expectancy still in the 50s and 60s. You might have died of a heart attack from eating all that red meat and working too hard without a vacation. Might have been HIV. People really couldn't tell the difference. Right? But you had about 15 really good years before stuff started to fall apart again. And so today, I think we're probably given up on most new antibiotics. Levaquin, probably one of the newer ones. Vancomycin is not working on certain stuff. Carbenipin resistant K pneumo is totally a thing. So with drug companies not making money or making their money back, I should say, before the microbes become resistant, what do we do next? Is it vaccination for every bacterium? Is it back to the bacteriophage? Do we use targeted proteins? And for now, the answer is just yes, all of those things, right? Excuse me, we have to see how this stuff pans out. And so that's kind of why we're in limbo now. We've got mRNA viruses that we've never had and mRNA vaccines that actually work. We've got new drugs that we've never seen before. We're going back into the banks and seeing if any of these classically, historically awesome drugs work today. And we've had some good hits. There was a new drug I just saw, and I should have printed it. It's 20 times more potent than Remdesivir in treating COVID. It's also way more expensive, but it's something old that now became new again. So it's not all doom and gloom. There definitely is promise as we use technology and DNA tech. But obviously, we want to do this in a safe manner. We don't need more examples of the Salk vaccine happening, especially right now with COVID. If that happened with any of these vaccines, I would be deeply concerned for the trajectory of this virus and the pandemic in the world. So we've used a lot of words. I'm going to kind of break here for a quick second here, see where folks have some questions. Probably not going to bore the daylight out of us for another hour, but certainly let's get us to a good point where we can stop. What do you guys think? Scientists seem pretty easy. Hopefully the history wasn't just an exercise. You've heard some of it probably somewhere. And there's so many more examples. Those are just kind of highlights. So many other people, so many other scientists doing so much other work. And the one thing that no one ever questions me about, and shame on me, so I'm going to throw this name into the chat. What did you notice about all the scientists? Nobody noticed anything interesting? Exactly correct. They were all men. But obviously, folks know about Rosalind Franklin and her role in the discovery of DNA. 1940s, didn't get the Nobel Prize because she had died beforehand. Rebecca Lansfield is a more modern scientist. She lived until the 70s, was the chair of microbiology, I want to say Columbia or NYU. It was local. And she discovered the Lansfield groups for Streptococcus. Right, so there are a lot of contributions. But unfortunately, a lot of these are overshadowed. And so I will do better in my due diligence, but I did want to bring that up and I did want to draw attention to that because there are some really good scientists. It should not matter gender, creed, anything. Achievements are achievements, and that's something that's just going to have to change in the world, isn't it? Maybe we're doing a good job, maybe we're not, but credit where credit's due. I'm a fan of that. So that's in the chat in case you want to look that up. That'll come back in your labs as we go. So let's see, what do we have? Some of that, some of this. Yeah. Okay. So the only other piece that's somewhat critical to kind of talk about this evening, I would say is going to be a piece of taxonomy. And I'm sure taxonomy, everybody's probably been exposed to it through the Roadrunner and Coyote cartoons. Y'all ever remember watching those on Saturday morning? And they would always do a quick freeze frame and you'd see like hungriest, ravenous underneath the coyote. Total bullshit. There's no such genus or species, but it was describing the coyote, right? And Speedius Maximus, like Speedius Maximus, what is that? Well, the Roadrunner was supposed to be really fast. In reality, the coyote is actually faster than a Roadrunner, so don't believe everything you see. But we were exposed to proper scientific notation. And I'm just going to throw, I can't throw because I can't do italics in here. All right, we'll do this. We'll think about taxonomy from a different perspective in terms of where we went and why we do binomial nomenclature. And I think that'll be plenty for the evening. So taxonomy is really the science of groupings. And unfortunately, human beings have a tendency to put things in boxes or silos. It's a very guilty pleasure. And it's something that unfortunately has been abused over the years as well. But it's kind of human nature to put things that are similar together and put things that are different away, and then put those new things that you find different with those other different things. Terrible description. But folks like Darwin did just that, right? Through the HMS Beagle expeditions, he put things in jars and he put like things together and dissimilar things away. And so what they really started with was the science of groupings that really wasn't formalized until Linnaeus came along in the later 1700s. And the reason why Linnaeus was such a big deal and why he gets credit, not some of the earlier folks, is because there were descriptions and there were classification schemes, but they were only useful for local type folks. They were descriptive, they used native languages. And if I said to you, oh, turn right at the blue house, I'm going to be the first one to claim my wife tells me I have no business telling people anything about color. If I tell her it's a blue shirt, she tells me it's gray. If I tell her it's green, it's blue. The point is it's a nice shirt. It doesn't matter what color it is, but it's lost on both of us, right? And so the other problem is science is about communication. You've got to be able to tell others what you learned, what you found, how you found it. And let's face it, there are groups of people who do not like to speak to each other. Classically, we're going to go English and French here, right? The English don't like the French and vice versa. And the French move heaven and earth to use English words. And so if you're at Woodbury Commons or you're riding the train, you'll often hear people speaking Greek or Chinese or German or whatever, and an English word flies out. Computer, right? Because that didn't exist in ancient Greek times. Well, the French made a word for computer. They would not use the English word, no way, no how. And so when you have communication barriers, it's a big problem, right? No one's going to prosper, no one's going to learn, no one's going to earn. So Linnaeus tried to unify all of this by using Latin. And the beautiful thing about Latin is today, nobody uses it, right? So it's kind of the language of science, but it provides two things that descriptive native languages don't. Typically an exact description, although with some things like Staphylococcus aureus, that might be debatable. It's mostly a good description. And it provides us with a hierarchy. So we can group things generally, right? In kingdom Animalia, we know that those are animals. We class Mammalia, we know that they produce hair and milk. And so the hierarchy allows us to know something about these organisms, but when in reality, we do fall back on binomial nomenclature, right? A genus, capital, species, lowercase, naming structure, where Staphylococcus aureus tells us it's a small circular organism that grows in clusters like grapes that produces a golden growth. That's what Staphylococcus aureus exactly describes. Right, a small circular critter, it grows in clusters like grapes, and it has a golden growth or golden appearance to it. So it exactly tells you what to expect if you have Staphylococcus aureus and if you're growing it in the lab. And so a lot of these other organisms, Salmonella, right? There's a Salmonella typhi and there's a Salmonella typhimurium. Same genus, different species. So we know something about it from the genus name, but obviously they do a couple different things, and which is why they're different species. And the whole definition of species is a place we could argue pretty much all day long. But if we start, thinking about this, obviously Whitaker's Five Kingdoms, I'm sure you've heard of. Is that fair to say? Yeah, kingdom Animalia, Plantae, Fungi, Monera, and Protista, hopefully. And the idea of kingdom, phylum, class, order, family, genus, species. King Philip climbed over the fence and got shot, or whatever you'd prefer, right? So we know about the hierarchy, and it's kind of interesting how science has built something around something that humans do already. So certainly we know about kingdoms. There's 5 of them. I'm not sure how many folks know about the idea of a domain. Is that something that's familiar to folks, to any, to many, to most? A little head shake in there. I'm just kind of flipping through the screens. So the domain is an interesting thing, and it doesn't work exactly the way kingdoms do. They are somewhat broader, but they are somewhat not as broad either, and that's terrible. I'll just explain it. So kingdoms, and in particular, the Monera kingdom, started to cause problems for researchers in the 1970s. And so a scientist by the name of Carl Woese was looking at a bunch of RNAs from small single unicellular organisms, right? So monerans. And when he was looking at his DNA sequences, they were rDNA in particular, so RNA sequences, he noticed that the overwhelming majority of bacteria, Staph, E. coli, were very similar, 99% similar to one another, as they should be. But there was a small group of weird stuff, later to be found methanogens and stuff that lives in acid drainage mines. Deinococcus radiodurans lives in a nuclear reactor. He found that their rDNA sequences were completely whack. They were like 70% different than the established. And so as he stared at his X-ray films and as he started to do the biochemistry on these, he named them archaea because they looked archaic. In 2000, there was a 60-page paper that actually rearranged the tree of life. And we still acknowledge that bacteria came first, but archaea now are recognized as being different than bacteria and more in line with eukaryotes. And they know that because of the biochemistry. So we're expecting domains to be even more broad than kingdoms, and they are with respect to eukarya. So domain eukarya is really kingdom, protista, fungi, plantae, and animalia. But domain archaea is really a halving of the kingdom monera, because monera are all small unicellular single-celled organisms. The domain breaks the archaebacteria out separately from the eubacteria. And so it's a little confusing because it doesn't exactly match the hierarchy, but it does give you a little bit more information at the domain level. An archaea is slightly strange. It lives in extreme environments. Often they're considered extremophiles, and they do some really weird stuff. So when I say methanogens, you think methane. I would say when you think methane, you should think the Bermuda Triangle. There are massive, massive deposits of methane clathrates at the bottom of the ocean. and archaebacteria are responsible for producing them and using them. And when these massive clathrate shelves break, they aerosolize methane gas. And if you're trying to fly through air, no problem. Methane is nowhere near the density of air, so airplanes lose their lift and they crash. If you're a boat and you're suddenly hit with a bubble of gas, gaseous seawater is not buoyant. Your boat is going to go down like a brick. And we know this from the North Sea oil rigs. When they hit gas pockets, they too sink. And it seemed to make a lot of sense since there's a lot of this methane in the Bermuda Triangle. It's really the fault of the microbes, not the boogeyman, not aliens. Well, probably not either of those, but most likely it's the organisms at the bottom of the sea. And so that's kind of an interesting thing. Kingdoms we're good with, phyla we're good with, class, order, family. Notice genus is capital, being a noun, species being an adjective is lowercase. And so that's why genus is capitalized and species isn't. And if we were to write Staphylococcus aureus, the S in Staphylococcus would be capitalized, the A in aureus always lowercase. And if you're doing this by hand or typing, it should be underlined or italicized, right? And I'll show you that again, but that's why the genus is capital, it's a noun. The species is typically an adjective, it is lowercase. Now here's an example of using a species that we know as the wolf. Basically kingdom Animalia, so great, we know it's an animal. We know it has a backbone because it's chordata. We know it produces milk and has fur because it's a mammal. We know it is a meat eater, right? We know it has particular dental structures with the Candidae. And its genus is Canis, very much like Canis familiaris, your house dog. But the lupus denotes that it is not a house dog or a domestic animal, that it is in fact a wild animal belonging to the wolf. And so we would never say Animalia, chordata, mammalia, carnivora, Candidae, Canis, lupus. We just throw out Canis lupus because if they're in the same genus, they are automatically in the same family, order, class, phylum, and kingdom. And so that's kind of why we can get away with abbreviating Canis lupus, or if you've used it in a paper before, C. lupus, right? The Canis can be abbreviated. The convention is only on the second go around or subsequent go arounds. Let's see, what else should we talk about here? Yeah, I suppose this is probably where we should go. So the idea of binomial nomenclature, or using the genus and the species, is the formal procedure, the formal name given here. And there's some good examples beneath: Staphylococcus aureus, Bacillus subtilis, Escherichia coli. I definitely never write it out, otherwise I'm definitely have to copy and paste it. But these are typically how they'd be written. These are typically what you would see. In the labs, you'll probably get a spattering of maybe 12 to 15 microorganisms. Aspergillus niger, Corynebacterium, I think it just says species because they substitute all the time. Streptococcus bovis, Staphylococcus epidermidis. And as you guys read, you're going to find that some of these things that used to be called Streptococcus have been renamed, right? And it's for the same reason, you know, that we kind of talked about earlier in terms of, well, people make mistakes. The scientific method just proves a hypothesis correct. Doesn't mean that it's true. And so, believe it or not, in 1984, there was a huge reorganization of names, genuses, and species. And one of the things that you guys encounter frequently, look at your yogurts, look at your milks, you see Lactococcus lactis, or Lactobacter, or Lactobacillus in part of the probiotics. Well, prior to 1984, all of those were Streptococcus lactis. Right, so those cheese-making microbes, those dairy-using microbes were actually members of the strep family. And when the advent of microbe and molecular biology coming together, we found out we kind of did things a little dirty, and we had to shift gears and we had to reclassify them. So kind of a bummer, but you know, bound to happen, right? And so the idea of a species then probably is a part that does make us a little crazy. We've always been taught that it's species who can reproduce with each other and make a sterile offspring. Kind of hard to think about that, though, when you get to the bacterium or you get to the viral level. And so we've kind of fallen back onto a combination of structure and function similarities, but when you look in a scope and you see a little green ball, I don't know what the hell the structure is. I don't have a good microscope. I can't tell. I don't know how to further characterize that. So you can fall back to chemical fingerprinting, which is a workaround to get at the genes. Or we can just sequence the genes and find out what it is. Now, the problem with gene sequencing is sometimes you get really high similarities, but sometimes you don't. So for example, if every human being in this room had our DNA sequenced, we would probably be somewhere about 85 to 90% similar to the human genome. Now, I don't think that means we're not human. I think that's just an artifact in how the Genome Project was done. We took 30 humans out of 6 billion and we sequenced their DNA. 30 out of 6 billion. Billion. So yeah, that's not a representative sample, is it? Yet, if we look at particular genes in our bodies, if we look at those same rRNA or rDNA genes, we will be 99.997% the same, all of us, you, me, everybody. If I look at your urease genes, we might only be 70% similar, even though we are, in fact, Homo sapiens. We are the same genus and the same species. And so this really gets a debate going in the scientific community. Some of it is slander. Oh, those molecular biologists, kids are clowns. Oh, those old timers, they just don't get DNA. And like everything, the truth is somewhere in between, whereby you probably need to grow things in the lab. You probably do need to look at structure and function similarities, but the genetic analysis and the chemical analyses of metabolism are right up there with figuring out how things have changed. With COVID, there's what, 12 different viruses now that do exactly the same thing? They all have these random polymorphisms, these stupid mutations in key areas that make them more virulent, or that make them more sheddable, or that make them more transmissible. Nowhere have we ever seen one that makes it less deadly, right? And almost we never pay attention to those. But I will say this, and this is a hope, a ray of hope amongst the clouds. Viruses that kill high amounts of people or that are this virulent are typically not meant to be in us. And as it transfers around from person to person more and more, the mortality rate will almost always decrease because a dead host is a useless host. If you look in history, smallpox was higher, it fell to 30%. If you look, Ebola was almost 100% in the 1976 batch. Today it kills 35%. Right? That's 40 years of passing between humans and bats back to humans and bats. So, you know, that's time and change. That's kind of evolution. That's kind of what happens when a virus jumps where it doesn't belong and we have to deal with it. And that's kind of where we are now. We're seeing COVID. I don't even know how many million people in the world have been infected, but we're definitely seeing mortality rates come down. We're seeing people react better to it. The doctors are better at it. And so we're not out of the woods, but things are in a more positive direction. How about that? And so looking at what's left, it's basically the scientific method in evolution. I don't want to beat that up tonight because we seem fried already. It's been a good 2 hours and change that everybody's been more than willing and flexible to participate. So I'm going to hit stop on the video. I'll ask if there's questions. And next time, we'll briefly look at scientific method. We need to do it anyway. And we'll talk about time and change, although I think we've done a good job with it. Maybe I'll save that until we get to antibiotic resistance, because it's going to come right back up there again. But the scientific method, I definitely want to touch next time. Before we hit microscopy and